Yeah, as Rahul said, thank you, Rahul, for inviting me here. Uh, I'm a statistician by training. I worked in genetics institutes all throughout my life, and I do population prehistory. I'm going to walk you through a little bit of what we know about how humans have, by humans, I'll mostly concentrate on anatomically modern humans and what have our history been. So I start with where Monjari left, is that nothing to us makes sense except in the light of evolution. And to do this, I'd just to be on the same page, because I'm not trained in biology, which most of you might be. I'd like to be in the same page of the minimum of biology and genetics that we'll need for the rest of this talk. So this is a human cell. This is stolen from the internet. This is what we call as a nucleus. Within this nucleus is where the DNA lies. There is some amount of DNA in these mitochondria as well. Mitochondria are remnants from long back, of bacterial remnants from long back. They also contain a little bit of DNA, and this mitochondrial DNA is very little compared to the amount of DNA that we have here in the nucleus. Is the pointer working? Can you see? Okay, never mind. Behind
you, there's a stick that you can pick up.
Oh, okay. No, never mind. So the mitochondria is also very specific, because the mitochondria passes from the mother to the offsprings. So my mitochondria and your mitochondria is only from your mother, so that makes it a little unique in itself. All right, so the DNA is the most famous helical structure in the world. It has 4 letters, as we know, A, T, G, and C, and it's a fantastic organization. So it's organized in a helical structure. It's then coiled. It's coiled in a specific way into all of these chromosomes. And as humans, we have 23 pairs of this. 22 of them are autosomal. 1 is a sex chromosome. Our sexual identity is primarily because of these pair of chromosomes, and it's pairs because we get one from our father, one from our mother. Just to give you a sense of how big this is, so the cell, as you know, is microscopic. You cannot see a cell without the help of a microscope. And within the cell, there is this DNA. If you uncoil this DNA and stretch it out, that is about 6 feet. All right. So this is a wonderful, wonderful way in which this is packaged into this small thing. And there are 3 billion of these base pairs. 3 billion is just a huge number. So just if you imagine, if your 6-inch scale is 500 bases, then 3 billion is the kind of distance from Chennai to Kolkata. All right. So that's how big the DNA is. And if you compare the DNA of 2 individuals, like you and me, any 2 randomly selected individuals in this world, then we are 99.9% identical, which means that there is only 1 in 1,000 places where we are randomly different from each other, which means that we are hugely identical when we talk about percentages. But if you think about the number, because 3 billion is a large number, 0.1% of 3 billion is also a large number. So all of these variability, genetic variability that we have between individuals is essentially because of this 0.1% difference. Again, A, T, G, and C, they are organized. It's a sequence, and this is how it looks like. This is essentially a small position in our genome. This is a real data. This is one of the regions where we see a lot of variability. Just having one of this is kind of enough, in a sense, because as you imagine, 2 individuals in this world are 99.9% similar. Your father and your mother, or any of our parents, are 2 unrelated individuals. So on an average, they are 99.9% similar. So on an average, there are only 1 in 1,000 places where they differ. So if you have something like a reference sequence, right, a reference human sequence, then everybody will be equal to the reference sequence at most places. Only there will be some places where they will be different. In some places, they will be different. Both of them will be different from the reference. In some places, one will be the same as the reference, the other will be different from the reference. So if you imagine that the white is a reference, then your genome can be summarized into just these places where your father and mother has been different, or both have been different from the reference. So as you can imagine, I mean, you do not need to study all of these 3 billion bases to study an individual's genome. You can only look at these variant positions if you know where the variant positions lie, and that gives you a good sense of what the information content of your genome is and what the uniqueness of that genome is. So that's what I do. That's what all of us do, many of us do nowadays, that this is what serves as our data. And we try and extract information out of this. Just to give you a sense of some of the misconceptions about genes that we have. One misconception is that generally what we think about when you start thinking about a mutation, we think that a mutation would mean that something which was working is not working anymore. We do not always get diseases because of that. Diseases can happen because of excess of something. This is an example of a condition called Down syndrome. You might have seen babies like this, wonderful babies, very, very lovely, loving. They oftentimes have very dangerous, they have different kind of developmental delays and physical problems as well. They are caused because of an excess of something happening in chromosome 21. It's not always about diseases as well. If you clasp your hand like this, you'll find that one of these, putting your right on the left or the left on the right, one of these will make you a little bit more comfortable. If you just clasp your hand, you'll feel that there's a difference in the way you feel by clasping your hand. This is something, a trait which is probably has nothing to do with natural selection, doesn't give you any kind of advantage, selective or reproductive, but has a genetic basis in this. Similar things with rolling your tongue. Some of us can roll our tongue, some of us cannot. But rolling our tongue, is a simple kind of diversity which we have in all populations across the world, and there's probably nothing to do with any kind of natural selection. So what I mean to say here, convey here, is that there's an enormous amount of diversity which probably cannot be immediately related to some natural selection happening in, or related to natural selection or sexual selection for that matter. And because there is this amount of diversity which is neutral, so to say, there is a lot of these which do not bring any change to our fitness or ability to reproduce or survive. These are the ones which we use to try and decipher a lot about how we have moved around. Okay, so it all started with somewhere in 1963, and genetics at that time was very young. People were looking at proteins essentially. So people could painfully look at proteins, and they would look at structures in proteins, and this is about cytochrome C. And this lady, Margolish, she looked at proteins of different different species, and found that if two species are connected, phylogenetically connected in evolution or morphologically similar, their proteins look similar. If their species are distant, their proteins also look distant. So that's what she observed. And she apparently tried to theorize this, saying that maybe there is some kind of a clock ticking, in a sense that the number of changes you observe between two which has diverged for, say, last 500 years or 500,000 years will be like 1/10 of what if they have diverged 10 times back. So there's some kind of a unitary method. If they have diverged from a common ancestor in X amount of time, then what is the number of differences you observe if somebody has diverged for Y amounts of time? So this is how they looked at it. So suppose here is an example of a case where there will be two species here. This is they have diverged from a common ancestor. And if you think about your time as your y-axis, then this divergence is much more recent compared to this divergence. And if we put ourselves in perspective, this is me, and if we think about this, this is one of my closest living cousin around as a species, a chimpanzee, and whatever people tell you, a cow is relatively distant from all of us. So this is a way we start looking at it. So if the molecular clock hypothesis is correct, then between a chimpanzee and myself, I'll find a fewer number of differences in terms of genomes. Between a cow and myself, we'll find a larger number of differences. And that's what essentially what we find. Now, think about this. I mean, all of these phylogenies, all of these trees were drawn using something else. People have used some other kind of similarity or dissimilarity between animals to try and draw this phylogenies. And what we are trying to do is, when we start having molecular data, we are trying to see whether the molecular data fits with all of this. And what we find is it indeed does. So if you look at the sequences, this is how it would look like. If you compare between my genome and the chimpanzee, maybe we'll find one place where we are different. If you compare that with a cow, there will be many, many places where there will be differences. All right, so far so good. And this indeed did hold in a large variety of this. So there was this question of something which is that, okay, you see this broadly, but can you really calibrate to two real timelines? Which means that, so you know something like a mutation rate is this. A mutation rate is mu per whatever. Now, if you want to see that if you observe this many mutations in this many generations back or this many years, then can that be calibrated for a very long period of time as well? So how would you do that? So this was early days of molecular biology. The way people who do this is try to do it in comparison to this. That suppose again in this axis is the oldest fossil of a species that we have found. So this is the oldest baboon fossil that we have found. This is the old. So this black is the fossil divergence time. So this is the oldest fossils of the species that we have found, right? The black. The white we are trying to calibrate this using molecular divergence data. And the calibration point somehow here is calibration with cows, which is about 80 million years ago, right? So the oldest cow fossil that people have found is 80 million years ago. So this is using something else. This is using carbon dating. And then you think that the number of differences that we have with the cow is standardized according to that. So you superimpose that with your molecular divergence data, then calculate it accordingly.
Is this cow like cow modern cows, or is that
No, it's not. It's oldest evidence. You do not know whether that cow will be exactly like a cow that we have today. No.
But is it like a bison or a goat, or is it more close to a modern cow than this?
It is more close to a modern cow, but it's not a bison or it's not a bison. Yeah. But I don't think at this kind of these timelines that really does matter too much. I mean, they diverge so much in such a short span of time that probably that would not matter too much. But I mean, these were
like humans have existed for a few 100,000 years, so 80 million years looks like a huge timeline in comparison.
Of our divergence from the cows, right?
No, I'm saying okay.
So this is comparing our divergence from the cows, right?
Okay.
So you calculate the number of difference. This is the number. Yeah. So if you are trying to estimate it's actually twice that because you accumulate mutation in both the arms of this. So you calculate twice that.
I think that ancestor was probably not a cow.
It's not a cow. The ancestor is not a cow. Yeah. So this is the first evidence of a divergence where something like a cow has emerged. And so the humans emerged somewhere here. You're trying to calibrate where this is. Yeah, by looking at cows of modern times. And you do it for all of this. So the whole notion of molecular biology at that time was they were trying to prove that using molecular biology, you can get an evidence which is similar to what people have used, people have gotten using from other disciplines. One of the reasons, well, so what you found is generally it worked okay. For some of these real ancient animals, the molecular divergence time was way ancient compared to the fossil divergence time. And it was very interesting in a sense that what happened over time is people started to get more and more new fossils. So as you can imagine, this is contingent on the oldest fossil of shark that people have found. Now, if you discover a fossil of shark which is farther ancient, then you can actually get closer to the molecular divergence time. And as it happened, that people found that people started finding newer fossils and got closer and closer to the molecular divergence time. So this game has now completely reversed. Whatever people have drawn, whatever phylogenies people have inferred using all of these disciplines are now being redrawn using molecular biology. One of the greatest advantages of molecular biology, as you can imagine, is you can never claim that I have gotten the oldest possible shark fossil on this earth. I mean, nobody can claim that. But if you look at data, if you think that more or less there is a molecular clock, you can generate more and more data. Your understanding of the genome becomes more and more more and more enriched with more and more information. You can have better models to fit on this, and you can also put on this other kinds of evidence that you have about the geology, about the climate, about the geography of the planet, and put up all of this and put into your inference and come up with an inference. So this is something which has changed a lot. And one of the reasons it has changed is, of course, that we now got a lot more data. So what people start asking now is, now that you have got data, can we come back to asking questions where people are much more closely related? So you remember the earlier one where I showed that where there were different species at these arms? So can I now look at data and try and reconstruct the relationship between all of us? Or can I look at the data of my genome and your genome and reconstruct who our common ancestor is? If I look into my mitochondria and any of yours mitochondria, can I try and come up with an inference of when our great-grandmother, some great-grandmother we had as an ancestor? Can we come up with a common ancestor to us? And in order to do that, we use the same kind of principle. Suppose these are two sibs. Some of you, if you're watching cinema, you might know who these guys are. They are offsprings to a mother. Of course, their common ancestor is just one generation back. And if you have somebody who's an African Masai Mara individual, that individual and this individual might also have a common ancestry, might also have a common mother, but that will go long back, right? So the basic principle is very similar. If you now try and relate that to their mitochondria, these two individuals will have very similar mitochondria, except for sporadic changes which might have happened in one of these individuals, versus this individual who's systematically different because there has been mutations which have accumulated during this lineage, this passage of time. All right, so this is the basic framework that we count mutations, we have a molecular clock, and that's how we go. All right, so DNA is looked upon as a book of history, and when we talk about history, then we talk about really how things have evolved. So this is 1975, and there was this paper which shook the world by storms. This is Mary Claire King and Alan Wilson from Berkeley. Mary Claire King, you might know, is also the person who found the BRCA2 gene for breast cancer, for familial breast cancer. So she came up with a paper in Science where she showed that the chimpanzee and the humans are as close relatives as, so they are like 99.5% similar in their DNA level. So they're very, very close. So the number of differences that a chimpanzee and a human have is only 5 in 1,000 sequence, 5 in 1,000 DNA sequences. So, so Alan Wilson's lab was essentially looking into all of this. So they got a chimpanzee sequence done. They looked at humans and chimpanzee differences. They also had samples from all across the world. So they tried to use the same thing to try and build and resolve controversies about human or anatomically modern human evolution in the world. And this is what came out of their first paper. This is 1987, close after this. And this, if you think about this, this essentially is a tree, all right? So you have to imagine this as a tree. This is like where the tree is hanged, all right? And this is one arm of the tree. Is just because this arm is really long that they have kind of folded it in this way, all right? So there are two things to remember here. One is that this branch length, as you imagine, this branch length is how far back the ancestry is. So this is like a y-axis. This is how far back the ancestry is, and these are just individuals on this. So what they found was interesting in a sense that if you have an ancestor here, there are two major branches, and in one of these branches, if you think about this, all of these were blue individuals, and these are all people from Africa. And the rest are all distributed in the other branch. So as if the entire world is on one branch, they coalesce to a common ancestor here. And the interesting thing is, these are individuals who are probably in small populations in and around African villages. Their common ancestry goes way back compared to the common ancestry of these individuals. So everybody in the world has some common mother somewhere. Everybody who's a non-African in the world has a common mother somewhere here. And these individuals, they have a common mother which goes farther back in time. And they might be all from the same village. So this was absolutely fascinating at that time. So they say that this is an indication of that it all started somewhere in Africa. We had our African Eve. We started in Africa, then we moved out, and that's the reason. We moved out in a small population to populate the rest of the world, and that's the reason that everybody outside of Africa coalesced to some mother here, and that mother is a sister of somebody here, right? So it will go farther and farther back if you compare this in Africans.
I think it's worth noting, because there's a lot of confusion on this, that it doesn't mean there was only one woman alive at that time.
No, it's yeah, it's.
Did not have the lineages survive.
Yeah.
One had the lineages survive.
Yeah. So this is what they then did. This is using something which is called a hypervariable sequence one. Again, something which probably would not contribute too much to the selection of an individual or a species. And when they did this, if you look at this open white points are Africans, and the closed points are all non-Africans. As you see that the segregation is much better now, and if you have an ancestor somewhere here, these are different African populations. They are the ones which branch out the first, and eventually up until this point, it's all African populations essentially, and after that you start finding the non-African populations. They did this here, and these are all African populations, and these are African populations mostly from Eastern Africa. And that's the reason that Southeastern Africa is oftentimes looked at the place from which it all started. The next one is when they did with the entire mitochondria. And when they did with the entire mitochondria, again, you find this very, very long branch lengths in all of these African populations. This vertical line separates non-Africans here and Africans here. And this is the point where all non-Africans essentially coalesce. So this is like an indicator of common ancestry of all of these non-Africans here. And that, of course, as you can see, has many, many more branches to go till we get to the point where it's anchored by an ancestor chimpanzee. So this is where your tree is hanging. And if you look at this, then the branching for the non-Africans start much later. All of these deep branches are from people who are from different places of Africa. All right. So this is one by which after this they could also calibrate this time. And what they proposed is not only is that we started from a small population in Africa, but if you consider this the history of anatomically modern humans in this earth, then half of the time we have spent on this earth has been only here. So the first half of this game was played only in Africa. Only in the next half that we have went on and populated and moved and colonized the rest of this world. The earliest of these studies were using, most of them were using mitochondria and Y chromosome. The great advantage of doing that is a mitochondria is the one which we inherit from our mothers. So if you go one generation back, it's only the mothers who have it. So if you go back further in time, then you are looking at the ways the females have moved. If you look at Y chromosome is from a male to his father, and from the father to his father, and so on and so forth. So this would give you a sense of the female lineage, male lineage across the globe, or the male movements. There is a concept of something which is, mitochondria is also interesting for other reason. So there are some regions on the mitochondria which mutate very, very slowly. It's very, very stable. It do not mutate at all, almost. And there are some which mutate very, very fast. So the ones which mutate fast are like the seconds arm in your clock. The one which mutates really slow is like your hours arm. So by one of them you can scale something like hundreds or thousands of years. And by the one which mutates very fast, you can scale something like 15 years or 25 years or, 30 years or, 2 generations maybe, 50 years. So this kind of calibration is also possible in the mitochondria. And based on this, people have defined this into haplogroups and haplotypes. And we have been also pretty lucky in a sense that if you think about haplogroups, which are the ones where it's very, very stable, then if you look at the haplogroups, then this is a haplogroup which is called an L1 haplogroup. This is defined by one specific very, very stable mutation that we see in our mitochondria compared to a chimpanzee. This is something which is called an L2 haplogroup. All of this L1 and L2 haplogroups you find only in Africa. This is exclusively found in Africa and nowhere else. If you look at this fast mutating parts of the mitochondria and calibrate this, then you see that both of these are much more ancient. And if you compute the time, it's about 192,000 years. So it's about 200,000 years that is estimated when people have arrived on this earth, anatomically modern humans. The most recent of this haplogroup, which happened on the kind of a background of the L2, is a L3 haplogroup. And every other haplogroup which you have here in the rest of the world are two close cousins called M and N, and they are very close to L3. So none of these L3, L2, and L1 are found out of Africa, but rest of the populations out of Africa mostly have M and N. I mean, almost everybody in India, well, not almost everybody, say more than 70% of Indians would carry something which is called an M haplogroup. And if you calibrate that back in time, this is about 72,000 years back, and this is about 67,000 years back. So not only were this, as you can imagine, this is a different way of looking at this, at a slightly different kind of data. So when people tried to do this, calibrate this using different kinds of ways, they found the same basic inference remaining. It just becomes stronger and stronger with different lines of evidence where we find that this is how the movements have been. This individual is the single Indian individual who is included in this study. So if you put all of this together and draw a map of how people have moved out, so it's about 100,000 years ago when people started moving out. This part of the land was populated very, very early. Australia was surprisingly populated surprisingly early. It's about 60,000 years back when Australia was populated compared to Europe, which was populated much later. And this part of the world is populated way recently. It's about 15 to 35,000 after the last glacial maximum, which means the last of these. Okay, so I'm going to skip this part here, but because the simplest models for population evolution would be like this: that if you have a small population, the population will grow in size, and when it grows in size, it put pressure on its environment, and that's the time some of them might might get divided into two populations. One thing to note here is that generally when populations divide, the resultant population will have a diversity which is less than the diversity of the parent population, right? Not everything is represented here. If it's a large sample, then it has a better representation. If it's a small sample, it has a much lower representation. So the diversity generally decreases as you go farther and farther in time. Now, if there is some kind of a barrier between this, which is like a reproductive barrier, so they are like isolated populations now, they would accumulate new mutations and grow signatures of their own. So the question is that there are genetic mutations which are happening here. There are mutations which are happening here. They are not exchanging their genes, and there is a parent population. The question is, from the data, can you infer, just given the data, can you infer which population the individuals belong to? And that's what people try to do, and that's what people try to do using their autosomal data. Mitochondria, it's probably will not be possible, but with autosomal data, this might be something which is possible. So this is how it is, that there is one population, it differentiates and it grows signatures of their own, and this becomes 4 different populations, notionally notable from its 4 colors. So it has its own signature. The question is, if I'm given an individual's genome from this population, can I find can I assign this individual back without knowing which population this individual has come from? There is a slightly more complexity to this added if you are studying populations in today, because it might happen that these populations have at some point of time diverged and gotten signatures of their own, but now recently there might be gene exchange, right? So they might be separated for hundreds of years, but in the last 2 years or last 2 generations, they might have exchanged genes, right? This is something which happens very, very often. For instance, if you think about the slave trade, the Africans who were separated from the Europeans for a very long period of time, and the Europeans went to United States, the Africans lived in Africa, they went to United States, and now there is some kind of an admixture between them. So can you find, if you are given an individual who is an African American, can you look at the individual's genome and say that this individual is 80% African and 20% European? So that's the kind of questions that people started asking. And this is something which is admixed, and this is what you can actually, and it turns out that you can actually do that. You can use nice mathematical models to try and do that. You can do that on the autosomal loci. So this is autosomal data. It's not mitochondria and Y anymore. It's autosomal. And if you look at one of the first studies where people have done this, there were 377 autosomal loci, which means that 377 positions on the genome. There were about 1,000 individuals from 52 populations across the world. And if you look at the genome and try to mathematically, without knowing which population, which individual came from which population, try to mathematically put them into different clusters and beans, the way to look at this figure is, imagine this as this 1,065 individuals. So this is like 1,065 vertical lines. Each line is an individual. So you are trying to paint the individual's genome according to where this individual has come from. It's like a signature. You're trying to find that this individual genome has this signature, which is distinct from this signature. If I paint the first signature as purple, I'll paint the second signature as green, right? So that's what people have done. And this is the cluster they have resulted. Note it, I mean, they haven't used any kind of population proxies. They do not know which genome has come from which population. And what they find is it optimizes at a scenario where k equals 6, which means that it apparently tells you that there are 6 major population groups in this world. And all of these individuals whom you find here, all of these, you cannot see them, all of them are from Africa. If you think about the second picture onwards, see the Africans are separated from the second picture. If you divide the world into 3 groups, the Africans separate as one group, and the rest are here, mixed here, right? So at k equal to 6, it kind of optimizes. Here are Africans. Here are all people who are from Europe, Central Asia, Southeast Asia, and a little bit of so these are Pakistani tribes. These are people from what we call is East Asia, West Asia, and Central Asia. These are people from East Asia. mostly populations, different tribal populations from China. These are two populations from these are two populations from Pacific Islands. They are Melanesians and Polynesians, and these are people, the indigenous people of the from North and South America. So this is where it kind of optimizes at k equals 5. It optimized at k equals 5. So these were the 5 populations that they think of. So the first one is Africans. The second is what we call is Eurasia, essentially Europe, Asia. This is East Asia, mostly China, Japan, and Mongolia and those populations. These are the Pacific Island populations, and these are populations which are from which are the more recent populations from North and South of Americas. Yes. Yes. I will give you an example of a different data set. So what happens here is if you go increase k, the next population which it identifies is a very, very isolated population which has signatures of its own. So it's called a kalash. So that's, but it's good that it kind of mathematically optimizes at this point. And the optimization kind of goes down a little bit when it starts identifying this. So it has an evidence from both ways that this is probably one way of classifying this. When your resolution is at that level, I'll show you that if you increase the resolution, you can break down this into much more systematic signatures which you find here. All right, so this is what they did with 377 autosomal microsatellite loci, and the data changes over time, the technology moves, and this is only a few years' time when they have done that with 650,000 autosomal single nucleotide polymorphisms. And what you find is the basic signature, it's now much more visible. So the Africans cluster here, the Americans cluster here, the Oceanian, the Polynesians and the Melanesians, they are here. The East Asians here. The whole Eurasia now breaks down into different kinds of ancestries. This is primarily Europe. This is Mid-East. Mid-East essentially is West Asia. So the Bedouins, the Palestinians, etc., the Druze, they are like admixed between population ancestries of Europe, a little bit from Central South Asia, and something which is an ancestry unique to these places, and then there is Central South Asia. So this was clustered into one group in the previous section. Now, if you look at a resolution which involves 650,000 SNPs, then this breaks down into different clusters here. So here are the 7 clusters. If you look at a population-wise phylogenetic tree, so then this is how it looks like. The basic story still remains the same, that here is some kind of an ancestry, you're hanging this tree by this. All early divergences in the human populations have happened somewhere within Africa. No, so the way it has happened is that this has been much more recent admixtures, where these individuals some of them have come back. So it has been like a melting pot. There has been migration movements which are much more recent. So after these ancestors have diverged, people have again come back and contributed their genes into this. So that is why so this is more a reflection of more recent admixture happening. These are all extant population today. You're reconstructing it all from today. All right. So what we found is seven major clusters. You can also identify admixture signals between populations, and geography is a wonderful correlate. If you start from any place in the world and try to correlate diversity according to a hypothetical path of movement around this world, you find that the correlation between genetic distance and geographical distance is maximum when you start somewhere around Addis Ababa and look and take the speculated route that people have used. So it's way more. That correlation is way more with the geographical distance and the genetic distance. There has been evidences, many evidences. I mean, there's, I will talk just of one more where people have looked into coevolution. This is coevolution of Mycobacterium tuberculosis, which is an obligate parasite. It only affects humans. And if you look at the Mycobacterium tuberculosis of lineages collected from different parts of the world, and if you put that in a phylogenetic tree, this is lineages from Africa, this is from Oceania, and so on and so forth. If you look at a modern day mitochondria tree, this looks like this, and the color codes are essentially people from the same continents. So as you can see, I mean, there is a huge correlation between the kind of trees that you observe between the way the MTB or the TB bacteria has moved along with humans in these parts of the world. So the migration is mimicked in the DNA of other organisms as well. So broadly, this is how it has been, that it started somewhere here. It has moved and, yes.
So the early African population themselves had TB?
Probably, yes. Probably, yes. I mean, carrying a bacteria does not necessarily mean that you have a frank disease, but they were carriers for sure. I think there is now evidence coming up that they must be having TB. I mean, they must be having a frank disease as well. So there was diversity in Africa. People moved around this for the 100,000 years people were there, and they moved to different places. And one thing to keep in mind is when we moved out, this was not vacuum. We didn't move out in vacuum. There were other hominid species, very surely, who were cohabitating these places, and some of them are very successful, like the Neanderthals. I'll talk a little bit about the Neanderthals. Neanderthals were very widespread in Europe when people moved out of Africa. But somehow, the last remains of Neanderthals that we find, last archaeological remains of Neanderthals that we find, is about 30,000 years back. Right? So the general idea is that we probably moved out and probably killed them or over-reproduced or whatever we did. The Neanderthals, we outcompete probably the Neanderthals after we moved out of, anatomically humans moved out of Africa. This is a much more real map. So if you can imagine, at those times, the seashores were actually much more extant, and the ice was trapped in these huge glaciers. I mean, this is one of the problems that we are now having, that these ices are melting and the sea level is rising. So at that point of time, when people started moving out 100,000 years back, there were a lot of these ice glaciers which held a lot of water, and that's the reason that the green is where the seashore today is. The brown is where the hypothetical seashores were at that point of time. So if you imagine people moved out of this, they could actually take a walk along the beach to get somewhere very close to Australia. So getting to Australia, which today looks so slightly strange to us, was not that strange, because you can actually go very close to this. Actually, you can go very close to the Wallace line and you can almost see Australia on a clear day. To get along this. And as you imagine, if humans have traveled through this, they must have attained some amount of boat craft to get along across the sea to Australia. So Australia, it was not that fascinated that Australia was populated so early. Anyway, so they moved around this, and no, the yellow are the Homo erectus migration routes. They were very similar to the Homo sapiens migration routes. This is not the first instance of hominids moving out of Africa after they have evolved somewhere in Africa. And these were probably extinct. Many of them, I mean, most of them were extinct before the modern humans moved out. But there were very clear evidence that there were some hominids who were still living there, like the Neanderthals. So I do mention the Neanderthals because that is going to be the next small part of my talk. So the question that was, so the theory that was proposed here is something which is called a replacement theory. So what it tells you that, yeah, the Neanderthals were there, we went and replaced all of them. And the question that people started asking is, can this be something like a slightly leaky replacement, that maybe it was not a complete replacement of all of this? So what is what essentially is our relationship with Neanderthals? Is there a way by which we can figure that out? I mean, if you look at the Neanderthal skeleton and compare it with humans, it's fascinatingly similar. It's just, I mean, they're huge. They're larger than what we are. They're stronger than what we are. So apparently you would think that they were successful, they were widespread, they were larger in number, they were stronger, they were bigger. Why would, and how on earth would we kind of wipe them off from this face of, I mean, we are moving into their territory in a sense when they were all across Europe. So how is it that we moved them all out? So the first part, a lot of this puzzle was solved by this person, Alan Wilson. Alan Wilson then started doing something which is, how much time do I have?
About 5 minutes if you can.
If I can. Okay.
We ended up starting a bit late.
All right. So would you give me 7 more minutes?
Okay.
All right. Okay. So this is a guy called Svante Pääbo. Svante had a very strange history in a sense that his father was a Nobel laureate. He was raised by his single mother though, and his mother his mother was an archaeologist. So he used to go to different places with his mother on vacation. And when he was a kid, he collected Egyptian mummies. Right? He collected a little bit of Egyptian mummies and kept it with him. When he was doing his PhD, he was working on the immune system during the day, and during the night, his project was trying to extract DNA out of this Egyptian mummies. And Alan Wilson was already very famous because Alan Wilson's lab has published all of these papers from mitochondria. They have revolutionized the world, saying that they have. The out of Africa theory was from his lab. Alan Wilson was also trying to do the same. And this is what Alan Wilson was trying to work on. So this is something which is like an extinct zebra. They found a little bit of remains from a zoo, and they tried to relate that. So this is an ancient DNA. This no longer exists. It's an extinct animal. They tried to find the DNA of this extinct animal. And what Svante was trying to do is trying to get the DNA out of a mummy, all right? And it somehow happened that they came up with their paper first in 1984, Alan Wilson's lab. Alan Wilson was not very convinced about his data. And then came Svante's paper. And Svante, he writes in his memoir that he was looking for a postdoc position in Alan Wilson's lab. And he was about to write to Alan Wilson, and after his PhD, he wrote the single author paper in Nature and got a letter from Alan Wilson. And the paper says that, dear Professor Pääbo, if there is a position in your lab, can I come and visit you for this summer? So Svante says, I'm sorry, Alan, I have to refuse you. I do not have a position in my lab. For that matter, I do not have a lab. So I'm going, if you agree, then we can reverse this offer and I can go and spend my time at your lab, which I was actually looking for. So that's how a postdoc arrived at Svante's home. And this is the excitement that it created right after these papers were published. Now, both Alan Wilson and Svante, they kind of agree that what they have looked at and what they said is the ancient DNA is not ancient DNA at all. So what they have done is a lot of contamination, et cetera, but somehow, and then at that time, people claimed that they have got ancient DNA from anywhere. The peak of the expectation was when they say that they have got ancient DNA from, you know what, right? Jurassic Park, from dinosaurs, et cetera. And there were really scientific papers which claimed that they have found the DNA of dinosaurs, et cetera. I mean, they have sequenced the DNA of dinosaurs. But I mean, they stuck to their pursuit. They tried to get DNA from Neanderthals, and they did this for a very long period of time, particularly in Svante's lab. The basic question to ask is, what makes us humans? So this is the cave from which they have collected these samples, and this is the breakthrough of the year 1997. So this science publishes the breakthroughs of the year. This was the breakthrough of the year 1997, when Svante really convincingly found DNA from the mitochondria of Neanderthals. And what they said is, so the question was, one of the questions which the people who oppose the out of Africa evolution or out of Africa movement of humans, who would say that the humans are so diverse in this world and the Europeans are actually descendants from Neanderthals, and then there you get a Peking man, and the East Asians who are so different are also like descendants of the Peking man, and so on and so forth. It's not like a replacement, but we are descendant of this so-called hominid species. They are our ancestors. People probably Europeans probably feel much more comfortable with Neanderthals than with Africans. But what happened was, in this paper, what they have showed is this, that they have compared they have found 123 clones from Neanderthals, and it was not a large data, 379 base pairs on the mitochondria. They compared that with some African and South European population, and this is what they did. So if you compare a human versus a human, there are a number of differences, and you count the number of differences and plot this on the y-axis. So the percentage of pairs that shows 5 differences between humans. So this is how the human distribution looks like. On an average, it has about 5 differences, right? If you compare a human with a Neanderthal, on an average, it has something like 25 differences. If you compare a human with a chimpanzee, that has somewhere about 55 differences. So we kind of know that this is 5 million years, right? So it takes 5 million years of separation between the human and the chimpanzee to get, on an average, 55 differences on that small part of the genome. So if you now do this molecular clock kind of a mathematics, then this turns out to be something around 1 million years. So they kind of, that paper kind of said that these are completely different species with no overlaps, and the number of differences humans have with humans, irrespective of where in this world you collect them from, is much lesser, significantly lesser than the humans have with Neanderthals, and that fits with the model of where the human chimpanzee divide is. The question is, the question is that the mitochondria do not recombine. You cannot recombine a mitochondrial DNA. So the question that remains is, while in the process of replacement, is it possible that humans actually did admix with Neanderthals, or anatomically modern humans did admix with Neanderthals? So that question kept bugging these people, including Savante, and at some point of time, in more recent times, we have a new kind of technology which is called next generation sequencing, which have made sequencing much more easier, and there is a huge amount of data that we can generate now. So what they did was, they could sequence 379 bases, right? And now, in 2009, they could sequence the entire mitochondria of a Neanderthal, which is about 16 kilobases. And in 2010, like you have a reference sequence for humans, they could have a reference sequence for the Neanderthals. So now you have a reference sequence for the Neanderthals. This went on. So this would enable us to ask questions like, okay, so you have a reference sequence for humans, you have a reference sequence for Neanderthals. Now if you take an individual's genome and compare it with the two genomes, are there positions on my genome where I match the Neanderthal much closely compared to how I match the human reference, right? So the question that you can now ask is, has there been early evidence of admixture between Neanderthals and humans so that some of these still remains today, right? So you can see that this can be done now, that there are these references, and if your genome at some places is like a Neanderthal, then you can imagine that that has come from an ancestor who was a Neanderthal. All right, so that's what they did. And what they found was that human and Neanderthals are not like two isolated airtight chambers. And if you compare between the genomes, yeah, it is true that the number of differences that you find between a human reference and a Neanderthal reference would give you a separation of 80,000 years back. And this is how the Neanderthal skeleton looks like. But if you compare the genomes of all people, then all non-Africans in this world is somewhere around 1 to 4% of Neanderthals. So in all our genomes, there is somewhere around 1 to 4% of a Neanderthal lineage. And amazing is that it's not the highest in Europe. The Neanderthal proportion of Neanderthal lineage is not the highest in Europe. In East Asia, it's higher. It's highest in places like here. It's way more higher than any other place in other parts of the world. And Africans almost do not have any Neanderthal admixture. So the model here is that people moved out. Yes, I mean the basic framework still remains. People moved out. When they were here, they admixed with the Neanderthals. There was a certain amount of admixture which you can estimate and which is pretty high. And then you move to different places of the world. And when you move to different places of the world, there were some Neanderthals here as well. And you continue to do that. And now we have much more evidence that you have actually continued to do that. So that when you have populated other parts of the world, everywhere, even after these guys got extinct, you could still find remnants of their genomes in us. All right, so it kind of reinforces the out of Africa model, but it's not a complete replacement. It's some kind of, there are leaks in this, and we have actually exchanged genes in that. So there has been a gene exchange. So I'll end this with a small story, two small stories. One is that this obviously, as you can imagine, would have been out of the ambit of any archaeological pursuit, right? Disciplines like archaeology would never find the possibility of an exchange of genomes or an exchange of genomes between Neanderthals and humans, right? I should also mention here that there is another twist in this story. So they were collecting Neanderthals from caves. Caves are wonderful because caves have their temperature kind of stabilized and the DNA remains intact. So there was this cave in a place called Denisova, somewhere in Russia, and they were Siberia, southern Siberia. They found one of the small bone from this finger of a kid, a Neanderthal kid. That's what they supposed. They did the sequencing of that, found a completely different species, a hominid, who is not a Neanderthal, not an anatomically modern human. They called this a Denisovan, because that individual is from a Denisovan cave. They called this as a Denisovan. Now, there are evidences coming out from genomic studies that Denisovans were also pretty widespread across this globe. And I'll just tell you two stories where this is a story of high altitude living. This is the evidence of the first state adaptation to environment that humans or anatomically modern humans have done. There are 3 places in the world where we can live really in high altitude. You know that there is problem living in high altitude because high altitude have reduced oxygen, and you and I would probably not live very long if we go there. So there are 3 places, one in Andes, one in Ethiopian highlands, another in Tibet. The Tibet is the one which is at the highest altitude. There are differences in physiologies in people who live in there. So the people who live in Andes and the people who live in Ethiopia, they have higher hemoglobin levels. People who live in Tibet, in the highest altitude in the world, they have hemoglobin level like you and me, indistinguishable from you and me. Actually, oftentimes much lower. And I mean, I do not know. If you go to the mountains, you know how these individuals are, what kind of workload these individuals can take. And this is because of selection in 2 very, very specific genes which people have found out. There might be other places as well. So the Tibetan highlanders live in very high altitude, but the blood hemoglobin is low. That helps them because a very high concentration of blood hemoglobin increases their chances of getting heart attacks. And the way they handle this is by efficient handling of the small amount of oxygen that they have. And this gene, EPAS1, is one of the genes which in the last 3,000 years have been the one which people think is the one which is responsible for many of this. And 40% of Tibetans, all Tibetans have this. Very few other people have these specific variants. And if you compare that genome is very, very close to what the Denisovans have. So the Neanderthals do not have it, the chimpanzee do not have it, no other populations have it, but this particular sequence of the EPAS1 is very similar to what the Denisovans have. So it is speculated that maybe long time back, this gene somehow integrate from the Denisovans into these people and enable them to live much more efficiently at a much higher altitude. So this is a paper which came out in Nature pretty recently, forgot which year. I'll tell you with another story to end. So it's about the human immune system. And the human immune system is very complex. It has many, many genes. One of the most important of them is this HLS. This is HLA class 1. Think about this. You are living in Africa. You are accustomed to the diversity of Africa. The world is changing. And you start moving towards Europe, all right? So you are exposed to a completely new kind of an environment. And the entire pathogen load changes, right? The pathogens that you encounter in Africa is very different from the pathogen that you are going to encounter when you move into Europe. And you need your immune system to be equipped to fight against them. And this is how we actually replace them. Wonderful usage of these tools. So this is one of those which is found in Neanderthals. And this is one which we have taken from the Neanderthals and the Denisovans, and this is one which has given us surely this has given us a higher immunity to live in a completely different kind of an environment. So it has actually been a replacement which has benefited us, and there are other evidences of this all throughout the human immune system where we have actually been benefited from our gene exchange with Neanderthals. All right, so I think that was my last slide. Oh no, not the last slide. Just one more thing to add to this, that this is how the tree now looks like. So there are the Neanderthals who moved out, the Denisovans moved out from the Neanderthals, and then they were also very diverse and they lived in different places. And this is where the human lineage differentiates. These are the Africans with very little admixture with the Denisovans, but we have admixtures with the Denisovans and the Neanderthals at different time points before they got extinct somewhere around 30,000 to 40,000 years ago. All right, so one thing just to note here is evolution is not about this. It's not about that nature is trying to design you as the best and the fittest and the wonderful and the most beautiful advanced animal in this world. Evolution is about a lot of these trials and errors where many people have many things which are better than what you have. And we have a species, we have just come into this earth. In terms of evolutionary fitness, we still do not know how we are doing. And there are many, many of them who have actually done pretty good. And they have lived for pretty long periods of time. But you do not see them anymore. And if this is timeline, then this is where we are, right? We are a species who have just started our journey. So it's better that we be careful about this. And this is something which we should also keep in mind. Let's be humble about this, that it's not that you are being designed to rule this world as the best and the last in evolution. It's a trial which is going on for a very long period of time. It will continue to go whether you and me survive or not to see that. We will obviously not survive in the very long scheme of things, but whether we survive even in the short scheme of things is our take. All right. So, thank you.
Questions? Or we could, okay.
Directly go to coffee.
Yeah, we'll have a very short coffee break for, let's say, 5 minutes. We'll come back here at 12:20, maybe 12:25 maximum. The coffee's right outside here. And then we'll move on. And lunch will be like 1:20. Sorry? You want to ask a question now? You can ask it.
I was just wondering that, of course, the common ancestor was in Africa. But what is so special about Africa? There are other tropical countries where why was nothing happening or was nothing significant happening there? Second thing was, is there nothing which is into Africa? Everything is out of Africa?
So if you think about this, that should I answer the first question first? First question. So the one reason why even Darwin was forced to speculate about, in Descent of Man, he was forced to speculate about human prehistory. And Darwin thought that somehow you see this large apes, this diversity of large apes in Africa. So common ancestry to humans, Darwin speculated, has to be somewhere in Africa as well. So in other places, you do not even see the large apes. I mean, there are large apes now. We know some of these large apes are there, but this diversity is somewhere not there. So the chances of human evolving out of Africa or hominids evolving out of Africa is going to be very low. So that was Darwin's take on this. I think nothing has changed too much about that. I mean, we do not have any more insight into this to say that something else is so spatial in Africa. About the second question, yes, there has been, but if you think about this, that once the modern humans have come into being somewhere in Africa, the African continent was such that, so this is all related to the way the geography has also, or the climate has also changed. So one reason why, so people have tried to figure out that what was so spatial, that about 100,000 years ago people started moving out. I mean, they were pretty good. I mean, they were pretty diverse. They went and populated different parts of Africa, which were habitable. So around this time point, if you look at geologically, there were a shifting in everything. I mean, so the places where they were living, those places were drying up, and there were some other places where, so I forgot the exact term for this. Other places were starting to mimic these places. So it was kind of a search for the identified niche, a search for the comfortable niche that where they used to live, and that's probably what has led them to move out of Africa. There has been movements into Africa after that, but as you can imagine, the first half, for 100,000 years, they were only in Africa, so the divergence was already so huge that there were some movements into Africa later times, but it's probably not so.